Bismillahirrahmanirrahim. Assalamu alaikum. This lesson is about differences between prokaryotic and eukaryotic cell. Here you can see this is an animal cell and this has a proper membrane bounded nucleus. Similarly, this is another cell, it is plant cell, and here you can see it has membrane bounded nucleus. This is a bacterial cell. These do not have a proper membrane bounded nucleus. Its DNA or its chromosome is present in a specific region which is called nucleide. So we will discuss about these two type of cells. The first one, this one is called prokaryotic cell, and this one is called eukaryotic cell. So pro means without, and karyon means nucleus. Similarly, u means true, and karyon means nucleus. This is the basic term on the basis of which we classify the cell into two types. The first difference between prokaryotic and eukaryotic cell is that prokaryotic cell do not have proper membrane bounded nucleus, and on the other hand eukaryotic cell has membrane bounded nucleus. Second difference, which is considered mostly important, is the presence of cell wall and its material. In prokaryotic cell, it is made of peptidoglycan. Generally, this is called as murin. On the other hand, in eukaryotic cell, if present, not in all cell wall is present. For example, animal cell do not have cell wall. If present, let's say for example in plant cell, it is made of cellulose mainly, and if it is a fungal cell, it is made of chitin. Chitin itself is made of glucose, to which nitrogen atom is attached. Next different is, this is also very important, the presence of ribosome. In case of prokaryotic cell, it has 70S ribosome, and in case of eukaryotic cell, it is 80S. S means Swedberg unit. This is the unit of sedimentation. During this process, ultra sedimentation takes place when the ribosome of prokaryotic cell reaches to the 70 speed of ultra centrifugation. It will settle down, and in case of eukaryotic ribosome, when the speed reaches to 80 Swedberg, it will settle down. It does not mean it's wet or something like that. Here you can see the detail of prokaryotic and eukaryotic ribosome. This whole structure from here to here, this is 70 s, means at the rate of 70 sedimentation Swedberg, it will settle down at the bottom of test tube, etc. Again, the larger part of this subunit, this is the larger subunit, it is 50s, and this one is smaller subunit, this is 30 subunit. Means this larger subunit, when present, it will sediment at the rate of 50 Swedberg, and this smaller subunit will sediment at the rate of 30 Swedberg unit. Again, here you can see this 5s RNA means it smaller piece of RNA with protein, which is the component of ribosome. This will sediment at the rate of 5s, and this larger RNA will sediment at the rate of 23 Swedberg unit. Similarly, this smaller subunit has only one RNA. This part is protein, and this is the RNA. It will sediment at the rate of 16 Swedberg. On the other hand, in case of eukaryotic cell, Here you can see it is ATS. This is the larger subunit and this is the smaller subunit. Larger subunit will sediment at the rate of 60 and a smaller subunit will sediment at the rate of 40 Swedberg. Similarly, this also has 3 pieces of RNA. This one, this is 5s, means it will sediment at the rate of 5s. This one at 5.8s, and the larger piece of the RNA will sediment at 28 Swedberg. Similarly, this smaller subunit has only one type of RNA, and it will sediment at the rate of 18s. Here, one thing should be clear, that it does not mean that 40 and 60 become equal to 100, and why it is 80s. On the other hand, here 50 and 30 does not mean that it amounts 80 if added. It simply means that if both these parts, both the subunit are present, it will sediment at 70. If the larger part is present, it will sediment at a 50 and smaller at 30. On the other hand, in eukaryotic cell, if both the subunit are present, these are joined together by magnesium ion, it will sediment at ATS. This larger subunit will settle down at 60 and this smaller one at 40 Swedberg unit. This is another main difference between prokaryotic and eukaryotic cell. This is, for example, bacterial cell. Here in blue you can see this one. This is the genetic material, which is also called as DNA or chromosome, and this dotted area, which is greenish in color, this is the region which is called as nucleoid. Here you can see there is no proper membrane bounding up around the chromosome or DNA. It is the region where its DNA or chromosome is present. This region is called as nucleoid. This is present in prokaryotic cell. On the other hand, this is, for example, animal cell, and this is the nucleus of animal cell. This is endoplasmic reticulum, this is mitochondria, etc. This nucleus has 2 layers, double membrane there, and it's inside this nucleus, either there will be present chromosome. For example, this is a chromosome, this is one, and this is another chromosome. Depending upon the condition, if the cell is in division phase, you will see chromosome this like structure, and if the cell is at interphase, then you will see such thread like structure, not the chromosome, these thread like structure. So when the cell is going to divide, these thread like structure, which are called as chromatin, it condense and contract and become chromosome. So in this case, there is a proper membrane bounded nucleus. This is eukaryotic cell. Next difference is that, that there is no organelles in prokaryotic cell except ribosome, which you have seen previously. On the other hand, eukaryotic cell has various organelles, for example, mitochondria, Golgi bodies, endoplasmic reticulum, vacuole, centrosome, plastid. Centrosome or centriole is present in animal cell, and plastids are present in plant cell. Next difference is the division of the cell. Frequently it occurs by binary fission. Binary means 2. The cell divides into 2 equal or unequal cell when conditions are favorable during 20 minutes. For example, pH, temperature, and food. On the other hand, the eukaryotic cell divide either by mitosis or meiosis. In mitosis, the number of chromosomes remain constant, and in meiosis, the number become half. Next, Actually the cell in this case in prokaryotic cell divides by mesosome. What is mesosome? For example, let's say this is a bacterial cell. In the center of bacterial cell there is a body. For example, here this is the body which is round about in the center of bacterial cell. This body divides ultimately the bacterial cell into two. This is called as mesosome. In this case, in eukaryotic cell, the cell either divides by middle lamella. Lamella means plate. This is, for example, in case of plant cell, this is a cell. A cell plate is produced in the center. This plate is called as middle lamella. In case of animal cell, it divides by cell group. For example, this is animal cell. Its division occurs by cell group in this way. Invaginates from one side and also from the other side, and it then divides the cell into two equal halves. Unfortunately, this is not working. This is, for example, one cell, and this is another cell. This is the region invagination. It is called as cell group. Next difference is the presence of extra chromosomal DNA, which is also called plasmid. It is frequently absent in eukaryotic cell, except some cell. This is, let's say, for example, this is a bacterial cell. This is the nucleide, and in this nucleide, here is the genetic material. It also has an extra chromosomal DNA, which is rounded in shape. This is called as plasmid. This is the property of prokaryotic cell. In eukaryotic cell, generally it is absent. Next is the actual time when the cell divides into two. Normally in bacterial cell it is 20 minutes if condition are favorable, and in eukaryotic cell it depends upon the cell division. It is 5 to 6 hours to complete the S synthesis phase, when DNA is copied from DNA, and then it is followed by 2 to 3 hours, and ultimately it takes 18 to 20 hours to complete the cell division. And here is the example of prokaryotic cell: bacteria, cyanobacteria, which is also called as blue-green algae, and archaea, archaea, you may like to pronounce. In case of eukaryotic cell, this is plant cell, animal cell, fungal cell, etc. And that is the end of this lesson. Thanks for watching, and it is hoped you have learned much more in this lesson.